Hello everyone, our topic is Herceptin, a revolutionary monoclonal antibody for cancer therapy. Firstly, I will talk about Herceptin's background and mechanism. Breast cancer is the most common malignancy among women worldwide. According to statistics from the WHO, approximately 2.3 million new cases of breast cancer were reported globally in 2022, resulting in about 600,000 deaths. HER2-positive breast cancer is a critical molecular subtype, accounting for approximately 15 to 20% of all breast cancer cases, leading to overexpression of the HER2 protein on the surface of cancer cells. This subtype is associated with higher malignancy and is more prone to recurrence and metastasis. In the late 20th century, monoclonal antibodies marked a breakthrough in target cancer therapy. In 1998, Herceptin was developed and approved for clinic use, becoming the first target drug for breast cancer. Herceptin binds specifically to the HER2 receptor, blocking the signaling pathways that stimulate tumor growth. The use of Herceptin significantly improves patient survival rates and reduces the risk of recurrence. Its introduction not only a revolutionary of breast cancer treatment, but also paved the way for development of targeted therapies in other cancers. Herceptin is structurally composed of two heavy chains and two light chains forming a Y-shaped structure. Herceptin have two main action mechanisms. The middle chart illustrates the high affinity binding between Herceptin and several accessible loop structures on the HER2 side chains, thereby prevent HER2 from combining with other receptors until they are internalized and degraded. And Herceptin can bind to receptors on immune cells. The immune cells release perforin and granzymes, leading to apoptosis of the target tumor cells.
Then let's talk about therapeutic application of Herceptin in HER2 positive cancers. Let's first talk about breast cancer. In this kind of cancer, amplification of HER2 gene leads to receptor overexpression, fueling uncontrolled cell growth and metastasis. Historically, these tumors had poor outcomes, rapid progression, and high recurrence rates. Herceptin changed this paradigm by binding HER2's extracellular domain. It blocks dimerization and recruits immune cells via ADCC. Clinical trials show dramatic benefits. Adding Herceptin to chemotherapy extended median survival by nearly 5 months and doubled the response rates. Today, it's standard in both early stage and metastatic settings, turning HER2 positive breast cancer into a manageable disease. Also, HER2 overexpression occurs in 15 to 20% of gastric and GEJ adenocarcinomas, particularly the intestinal subtype. Unlike breast cancer, HER2 expression here is patchy and localized to glandular surfaces, complicating diagnosis. The TOGA trials improve Herceptin efficiency here too. Combined with chemotherapy, it improved survival by 2.7 months and boosted response rates by 13%. This leads to its approval as first-line therapy for metastatic HER2 positive gastric and GEJ cancers with strong HER2 expression. In the end, Herceptin exemplifies precision oncology, transforming outcomes by targeting molecular drivers. Its success in breast and gastric cancers highlighting the importance of HER2 testing and tailored therapies, offering hope where prognosis was once dire.
So now I'll walk you through the production process of a humanized anti-HER2 monoclonal antibody, a vital therapeutic for HER2 positive cancers. The process spans 6 stages: genetic engineering, CHO cell screening, bioreactor optimization, purification, quality control, and concluding remarks. So now let's begin. The journey starts with genetic engineering. So first we insert the light and heavy chain genes of the antibody into a mammalian expression vector to ensure proper structural integrity. Simultaneously, we design the anti-EGR2 single-chain variable fragment and construct heavy and light chain plasmids. Screening markers such as antibiotic resistance genes and introduced to identify stably transfected CHO cells. This fundamental step guarantees precise antibody design and sets the stage for high-yield production. Next, we screen CHO cells for optimal performance using clone peaks imaging. We isolate monoclonal colonies with high expression levels. The clones undergo rigorous testing for HER2 binding activity and production stability. Long-term sub-culturing over 60 generations confirms genetic consistency, ensuring the selected cell line maintains high antibody output and functionality throughout large-scale production. In bioreactor cultivation, parameters are tightly controlled. The pH is dynamically adjusted between 6.8 to 7.2, while temperature shifts from 36.5 to 37 at the early stage to 33 to 35 at the late stage to prolong cell viability. Dissolved oxygen is maintained at 30 to 60%, and nutrients like glucose or glutamine are replenished using perfusion culture technology. The strategy boosts antibody yields to like 2 to 5 grams per liter, doubling traditional batch outputs while extending cell lifespan. So post-fermentation and purification begins, centrifugation and 0.2 milliamp filtration remove debris, followed by protein A affinity chromatography to capture the antibody's Fc region. After elution at pH 3 to 3.5 anion exchange and hydrophobic interaction, chromatography further refine purity. Viral safety is ensured through low pH incubation and nanofiltration. Finally, tangential flow filtration concentrates the antibody to 50 mg per ml and replaces the buffer for formulation. So last, quality control is paramount. Purity is validated via HPLC, SEC, and SDS-PEG. Functional assays include HER2 binding ELISA, cell proliferation inhibition, and ADCC-CDC activity comparisons with Herceptin. Safety thresholds are met, like HCP, DNA, and endotoxins. So in conclusion, the integrated approach ensures a high quality, efficient, and safe therapeutic antibody. So thank you. That is the end of my part.
Hi, my name is Yang Xin. I will briefly introduce bioreactor scale-up strategies, manufacturing modes, and cost control through biosimilar development. Fed batch is a traditional and widely used approach. It offers mature operation, simple training requirements, and is highly compliant with regulatory standards. Fed batch is particularly suited for large-scale production using bioreactors of up to 20,000 liters, which has been essential for blockbuster drugs like Herceptin. Perfusion processes provide higher cell densities and production yields by continuously feeding fresh media while removing waste. Perfusion enables continuous production, better resource utilization, and more stable product quality. It is increasingly used for flexible, small-scale, high-value production. When we compare fed batch with perfusion shows clear advantages. It improves yields, productivity per unit time, and the number of patients that can be treated. Perfusion is also linked to lower unit production costs, especially because of reduced facility investment and lower consumable usage. Speaking of cost control, let's focus on biosimilars. The introduction of biosimilars has substantially reduced treatment costs. On average, biosimilar treatment costs are about 40% lower than originator Herceptin, approximately $38,173. Moreover, biosimilar competition has led to a 52% reduction in overall treatment costs, with average saving of $45,659 per patient. Besides manufacturing improvements, administration route optimization also contributes to cost savings. Switch from intravenous to subcutaneous Herceptin saves 71.7% of preparation time and 89.3% of chair time. Subcutaneous administration becomes more cost-effective for patients weighing over 65 kilograms and improves treatment center efficiency, particularly where capacity is limited.
Hello everyone, my name is Xu Chenyue. Today I will talk about the challenge and the future direction of the Herceptin therapy. Let's begin with the current challenge. First, producing the Herceptin is complicated. You need advanced CHO cell engineering and the multi-step purifications which make it expensive. And we also need a better predictive biomarkers to find patients who will truly benefit. And then you can see the diagrams. It shows the major resistance mechanisms. And we find two are the most important. First is the immune variability. ADCC is weakened, and the tumor escape through the PD-1 or PD-L1 activation. And the second, molecular resistance like the HER2 loss and activations of the survival pathway. So the figures link the biological resistance to the clinical problems we faced. To solve those problems, we find some new strategy. First, improving the CHO engineering and continues the chromatography make the Herceptin cheaper and faster to produce. Second, as shown in the diagrams, combining the Herceptin with the PD-1 or PD-L1 antibodies helped restore the immune attack. Third, Herceptin is now being used beyond the breast cancer, like in the HER2 positive colorectal and gallbladder cancers. And finally, nanoparticle delivery systems are being developed to better target tumors and reduce the side effects. Looking ahead, the timeline shows how Herceptin keep evolving from the discovery to the biosimilar and then to now new strategy. Herceptin changed the precision oncology by helping the HER2 positive patients live longer and better. Biosimilars like the Oldrive make it more affordable worldwide. And in the future, technology like the nanomedicine and the personalized immunotherapy will help the Herceptin even smarter and more accessible.